Hello everyone, welcome. So today I'm going to talk about general principles of virology. Okay, so let's just start with basics of virus. So viruses are kind of very funny creatures, I should say. You can't call them cells, they are not cells. Because if you consider cells like a human cell, which is an eukaryotic cell, or a prokaryotic cell, especially an eukaryotic cell has a nucleus, a cytoplasm, mitochondria, and ribosomes. So you have the code, you have the field, you have the energy, and you have the protein synthesis machinery. So you have everything. So everything consists, everything are inside a cell. But in case of viruses doesn't have anything like that. So viruses are actually not cells, and your cells, like human cells, actually contains DNA inside the nucleus and different types of RNA in the cytoplasm, and actually both of them are present. But in case of virus, you get either a DNA or an RNA, but never both. That's an important difference between a normal cell and a virus. And as the viruses doesn't have the ribosomes and doesn't have the mitochondria, so it actually can't synthesize their own energy, and also as they don't have any ribosomes, they cannot synthesize their proteins also. And that's why virus has to infect a cell which has all those machineries, and virus actually use those machineries to replicate inside a cell. And that's why viruses are obligate intracellular organisms. And you can remember from your bacteriology, there are two obligate intracellular bacteria, which are Rickettsia and Chlamydia. And unlike human cells which go mitosis, or bacterial cell which undergo binary fission, viruses actually goes to multiplication or amplification. That means that one virus can actually create thousands or hundreds of viruses or progeny viruses. And in the surface of virus, you have something called surface protein, and these surface proteins are very important, determines the serotype, and also host specificity, meaning that which host is going to infect, and organ specificity, meaning which organ it's going to harbor. Okay, so those are the basics, very basics of viruses. So this is a picture that shows you a ball with some triangular shapes on its surfaces. Actually, this shape, which is actually a 3D structure, it's called an icosahedron. Icosa means 20 in Greek, and this globular structure actually has 20 circles. So 1, 2, 3, 4, 5, and about 20 triangles are present on the surface of this globular structure, and that's why it's called an icosahedron. And this is the most common viral structure encountered. So actually you see here, this is a virus. This is a 2D view, a cross section through the icosahedron. That's why it looks like a hexagon, but it's actually not a hexagon, it's an icosahedron. If you cut or transversely cut the icosahedron, you will get something like that. So this is a icosahedral capsid. So you know, the capsid is the outer portion of the virus. So outer protein portion of the virus is called capsid, and inside you have the nucleic acid, which is either DNA or RNA, but never both. And here you have a virus which doesn't have any envelope on the surface. So this is a naked virus, and the capsid shape is icosahedral. Okay, and now there are another type of virus which are called envelope virus, and I will discuss about that later. And naked viruses, they can either be DNA virus or RNA virus, but for all of them I have a mnemonic which actually I brought from First Aid 2015 edition. And it's suppose is situation you are in a clinic and a hippie came who is supposedly naked, and she needs CPR because her vitals are not good. And after giving CPR, you determine that as her age is about 28 or 30 and she never had a pap smear, now it's time that he came to the hospital or clinic. Now it's better to give her a pap smear, because you should be checking her on that too. So you have to give pap smear and CPR to a naked hippie. So pap smear and CPR to a naked hippie. So this is the mnemonic for remembering the naked or non-enveloped viruses. And for the pap portion, those are the DNA viruses, and the CPR and naked hippie, those are the RNA viruses. So for the first P, papillomaviruses, A, adenovirus, P, parvovirus, P, pneumovirus. So those are the DNA viruses. And below you have the RNA viruses, CPR, C for calicivirus, P for picornavirus, R for reovirus. And hippie means herpesvirus. And as the naked hippie, so it's showing you those all other naked viruses. And one very important concept to remember, that virtually all viruses that transmitted by fecal-oral route does not or do not have any envelope. And it's very fun to integrate this into your concept, because if a virus has an envelope, this envelope is actually very sensitive to different kind of environmental or body local situation. And if a virus is enveloped, if it goes through your GI tract, if it goes to the stomach acid will destroy it. So if an enveloped virus tries to infect the GI tract, it wouldn't be able to do so, because the envelope is susceptible to gastric acid destruction. Okay, that's why all viruses that are transmitted by fecal-oral route will be non-enveloped virus or naked viruses. Here you have the enveloped viruses. There are actually two types. Some have a icosahedral or icosahedral capsid, and some of the enveloped virus has a helical capsid. Here you have an enveloped virus with icosahedral capsid, and you have here is a membrane, and on the surface of the membrane you have some surface proteins. And as you can remember from the previous slide, it determines the serotype, the host specificity, the organ specificity. And this membrane is actually a lipid bilayer, and inside the membrane you have the capsid and the nucleic acid. Okay, and this is another type of enveloped virus with helical capsid. You see the inside there is the helical capsid. This is the helical capsid with nucleic acid inside, which cannot be seen from here. And also it has the lipid bilayer, and on the surface has a surface protein. Okay, and envelope viruses actually almost all of them acquire envelopes from plasma membranes of the cells that they infected. But there are a specific group which is herpes virus, which is a DNA virus, and as the DNA virus infects the nucleus, so it resides inside the nucleus and replicates there. And herpes virus actually when it gets out from the nucleus, it actually takes up the nuclear membrane of the nucleus. So the herpes virus has the envelope acquired from nuclear membranes. And as I have told already, that envelope virus has the envelope which is very sensitive to heat, drying, detergents, alcohol, and ether. So they are very sensitive. So they cannot stay in the environment for long. And that's why they have to be transmitted from one person to another person, or one animal to another animal, through direct contact of body fluid or by some other contacts. So you have to need a direct contact of transmission of envelope viruses. So it can be transmitted by blood transfusion, or sexual contact, or insect bite, or respiratory aerosol droplet. Very important to know. And here we are discussing about DNA viral genomes. So as you know, DNA in your body, you have the DNS, and it is deoxyribonucleic acid, and you have a double-stranded DNA helix, which is very important to know. So you have the double-stranded DNA helix. But in some viruses, especially in parvoviruses, this DNA can be single-stranded. So only parvovirus has a single-stranded DNA, but all other viruses has the DNA like us, meaning a double-stranded helical DNA. Okay, sorry, a double-stranded DNA, which can be linear, which can be circular. And parvas actually means small, and actually parvovirus is one of the smallest viruses. And the viral DNA can be linear like that, or can be circular like that. So most of the viral DNA are linear DNA, but some viruses which are papilloma, polyoma, and hepatinoviruses have circular DNA. Okay. So RNA viral genomes. RNAs are most commonly single-stranded in human body, and most of the viruses also have the single-stranded form of RNA. But one virus, which is reovirus, and one of the important reovirus is rotavirus. So reovirus group actually has a double-stranded RNA, and the mnemonic for it, repeatovirus. So repeated strands of RNA, 2 strands, and repeato, first portion re and then o, reo. So repeatoreo virus actually has a repeated RNA strand, or actually has 2 repeated RNA strands which are double-stranded RNA. And there is something called a positive strand RNA virus and something called a negative strand RNA virus. Actually, positive strand RNA virus doesn't need any polymerase for replication, but negative strand RNA virus need polymerase to convert to positive strand, and then they can replicate and they can infect. So there is a mnemonic for positive stranded RNA viruses, and the mnemonic is, you have to consider a situation. Suppose you are in California and someone invited you over to their party, naming a retro toga party, and you are going to that retro toga party, and after entering into the party you are offered something called a Corona drink, and when you taste it, it's very flavored. So it's a flavored Corona drink, and after drinking the flavored Corona, you also ate some pickles which were supposedly made by some hippies who also lived in California. Okay, so you went to a retro toga party where you drank flavored Corona and hippie California pickles. So retrovirus, toga virus, flavivirus, coronavirus, happy virus, calicivirus from California, and picona virus from pickles. So you can actually recreate the situation and practice it for multiple times to help you to remember it easily. And naked viral genomes can be naked viral genomes. Infectivity depends on what type of genome they are. If it is an RNA virus, it depends on positivity. If the RNA virus is positive strand RNA, it will be infective. If the RNA strand is negative strand RNA, it will not be infective. So it is not infectious. And if the DNA virus is present, or DNA virus tries to infect, if the DNA virus has purified nucleic acids, it will be infective or infectious. But naked nucleic acid of DNA viruses will not be infective. Okay, so especially the ssRNA require the polymerase contained in the complete virion, because the negative strand should be converted to positive strand if it wants to be infective or infectious. So viral replication depends on what type of virus are there. If it's a DNA virus, it would like to go along with DNA which is present in the nucleus. So most of the DNA viruses will be replicated inside the nucleus, except pox virus. And you have the RNAs in your cytoplasm. So most of the RNA viruses would like to go in the cytoplasm and replicate there. But there are some exceptions, which are influenza viruses, retroviruses, and hepatitis G viruses. So here we come with some viral genetics. What you can see here, you can see in the above panel 2 strands. One is 5' to 3' and another is 5' to 3' in different directions. Okay, so those strands actually now is crossing over, and after some time they share some genetic material, but a bit of genetic material. Here you can see the red panel or red gene and the blue gene, and you see the red gene is purely red and the blue gene is purely blue. But after the crossing over, the blue portion actually came here along with the red, and the red portion came here along with the blue. So you have a red-blue combination. So this is called a recombination because the combination is changed. So this is a recombination of the viral genome. And here you have something called a reassortment. So here you see the viral genome of one virus, and here you see viral genome from another virus. So this is the blue and this is the grayish one, and those are, you can see those genomes are fragmented or segmented genomes, and the segmented genomes can actually share their segments or actually transfer their segments between the viruses. And if that does happen, if you see the segments are actually transferred from one virus to another virus, and now you have a mixture of segments, this will be called a reassortment. Okay, this is very important for influenza virus pandemics. Okay, sorry, reassortment. So recombination is actually exchange of genes between two chromosomes by crossing over within regions of significant homology. And reassortment is if the viruses are having a segmented genome like influenza virus, the segments will be transferred from one virus to another virus, and this will be called a reassortment, and this will result in a very high frequency of gene exchange, and ultimately it can cause something called antigenic shift and leading to pandemics. And the 2009 novel H1N1 influenza A pandemic actually emerged via complex reassortment between human, swine and avian influenza viruses. And there is some other forms of viral genetics, which is complementation, and this is a very fun concept that you may have already know. And in here suppose two virus infects a cell, but one virus is defective, one virus doesn't have something to actually replicate, and the another virus has this particle, and the another virus which is able to replicate gives some particles which is needed for replication of the defective virus, and now the defective virus also can replicate like the normal virus. So the most common example is hepatitis D virus, which is a defective virus. It needs actually the hepatitis B surface antigen, so HBsAg, and HBsAg can be supplied by hepatitis B virus. So HBsAg comes over the hepatitis D virus and helps it to grow. So hepatitis D virus is a defective virus which is complemented by HBsAg with the help from hepatitis B virus. Okay, so this is complementation. So hepatitis B virus complements hepatitis D with HBsAg. So when one of two viruses that infect a cell has a mutation, so hepatitis D virus had the mutation that resulted in a non-functional protein, and the non-mutated virus, which is the hepatitis B virus, complements the mutated virus by making a functional protein which is HBsAg, and that serves the both viruses. So HBsAg is not only present in hepatitis B virus, but also it's present over the hepatitis D virus, and it helps the both viruses to replicate. Okay, so here is the hepatitis B virus is a helper virus, and hepatitis D virus is a defective virus. Okay, and there is another viral genetic change which is called phenotype mixing. So here the phenotypes are only mixed. Suppose this is virus A and this is virus B. Okay, and now you have some changes. Suppose those two viruses infect a single cell, and when a virus infects a cell, it actually get disassembled. So the viruses become broken down, the viral genome is released, and the viral genome actually is transcribed and then translated and forms new virus particles which are actually assembled again. So this disassembling and then reassembling can be somewhat complicated if the two viruses infect the same cell. So there can be some defects where the surface coat or the capsid of V virus or the envelope of V virus can get over with the genetic material of the A virus and vice versa. So here you can see the genetic material of the B virus covered with the surface proteins or the serotype markers or capsid of the A virus. Okay, so there is a mixture of phenotype, but genotype is the same. So you do not have any mixture of the genotype. So this is a pure genotype of the A virus, and this is a pure genotype of the B virus. But the surface protein, which actually determines the phenotypes, are actually changed, and that's why, as I have already mentioned, the surface protein determines the specificity, especially the host specificity and the organ specificity. So suppose this virus, as it has the surface protein of the B virus, it will infect the host and the organs that is commonly infected by the B virus. But when it infects that organ, the virus will again undergo disassembling, and then the A virus genome will be transcribed and translated, and it will produce the surface protein of the A virus. So ultimately, if this virus infects a cell, this virus will actually produce the A virus only, and the surface protein will now be actually the surface proteins that the A virus had. So after progeny, this virus will again be converted to this virus because it has the genomic information that creates these surface molecules. So this is the phenotype mixing. Okay. So here you have, so it occurs actually when simultaneous infection of a cell with two viruses occurs. Genome of virus A can be partially or completely coated, forming something called a pseudovirion with surface protein of virus B, and the type B protein code determines the tropism or infectivity of the hybrid virus. But if a progeny is formed that will have a typical type A code, and actually it was encoded by type A genetic material. So you can see here, so if it is a type A virus converted to something called a pseudovirus or pseudotype, and after a progeny virus is made, it will be again a simple, a pure A virus having the surface code of the A virus. So the progeny will have a type A code which is encoded by type A genetic material. So it will be the same again. Now we come to the vaccines. We have actually two types of vaccine. One is live vaccine, which is live attenuated vaccine, and another type is kill vaccine. So live vaccines have some distinct advantages over the kill vaccine, and one is it can induce both humoral and also cell-mediated immunity. But there is some problems. A live vaccine, the virulence of a live vaccine is actually reduced by some chemical or mechanical processes. But if you give a live vaccine to someone who is immunocompromised or a pregnant lady, this virus can actually revert back to their virulence. So can revert to virulence on rare occasions like immunocompromised patients or pregnant women. So that's why you shouldn't give live vaccine to those kind of patients. And another advantage is you don't need any booster, and you have actually two types of immunoglobulin produced. One is IgA and another is IgG. Okay, so you have to remember the common live vaccines that are commonly used in clinical practice, and for that you can actually fantasize a situation. Suppose you are in a tent and you have a poultry farm close to you, and actually you are shouting out leave, one not only, and you are telling another person who is beside you that can you see this small yellow rotating chickens which are getting vaccinated with Sabin and MMR? Isn't that incredible, vaccinating the chickens with Sabin and MMR? Oh, it's incredible. Sorry guys, it was not a good amount of fun, but actually you have to remember that. So the live attenuated vaccines are smallpox, yellow fever vaccine, rotavirus, chickenpox, Sabin polio, MMR, and the intranasal influenza. So live, small yellow rotating, the smallpox, yellow fever, rotavirus, and then the chickens. Sorry, I forgot to make that red. So chicken for chickenpox, and Sabin polio for oral polio vaccine, and the MMR for measles, mumps, and rubella, and incredible for influenza, which is the intranasal influenza vaccine. And the kill vaccine has some distinct advantage and disadvantage. The advantage is kill vaccine actually cannot revert to virulence, but the disadvantages are it has a shorter duration of immunity, it is less effective, and only IgGs are produced in comparison to live attenuated vaccine, which actually can produce both IgA and IgG. And kill vaccine actually does not produce or does not stimulate the cell-mediated immunity as much as the live vaccines does. So you can remember kill vaccines by this mnemonic: rest in peace always. So RIP always. R for rabies, I for influenza, P for Salk polio, and A for hepatitis A virus. And for Salk is killed. So the Salk is killed. So Salk polio vaccine, which is an injectable vaccine, is a kill vaccine. And there is something called a subunit vaccine, which are not a mesotype, which can be hepatitis B virus. Actually, it made from a part or a unit of hepatitis B virus, which is called a surface antigen, or HBsAg, hepatitis B surface antigen. And you can also have some human papillomavirus vaccine, especially against type 6, 11, 16, and 18, which are actually some parts of the human papillomavirus taken up, and those parts are actually injected, and they actually can build some immunity against human papillomavirus infection. And so you can get protection from CINs, cervical dysplasia, and cervical carcinoma. So that's all from me. Thanks for watching my video and stay tuned for more